This is the 7th lecture in Unit 1. In this lecture we will be looking at protein synthesis. This is one of the trickier things to learn, so don't be surprised if you have to view this section a few times. There are several animations to watch on here which are very helpful in understanding the process. Also be sure to ask questions if you need to, and be patient. You will eventually get this. There are two key processes involved with protein synthesis. In the first process, transcription, we will make a copy of the DNA that is stored in the nucleus and use it to make an mRNA. The DNA will serve as a template for building the mRNA. When the two strands of DNA are separated, only one other nucleotide or base pair is able to pair with any of the four used in the DNA on the single strand, so the complementary RNA nucleotide is automatically added. Transcription is the name given to the process of making the mRNA from the DNA, while translation is the name given to the process of making the protein from the mRNA. Here we will look at the key players involved with protein synthesis. The codon, or base triplet, is 3 base pairs that will code for a specific amino acid. This code can be translated using a codon table, which you will find later on in this presentation. The mRNA, or messenger RNA, will actually be read to make the protein. The tRNA, or transfer RNA, is what is going to be used to match the amino acid to the codon. The ribosomal RNA, or rRNA, is going to be present in the ribosome. This helps give it the ability to translate the code as it is part of the structure and read the instructions to build the protein. The promoter is a sequence at the beginning of the gene that will enhance protein synthesis. It is part of the regulation of protein synthesis, so we know which proteins to make when. The terminator is a nonsense sequence at the end that terminates protein synthesis. By nonsense, we mean that it does not code for an amino acid at all. The anticodon is the mirror image to the codon. This will be found on the end of the tRNA. In transcription, the double-stranded DNA is split apart by an enzyme in the nucleus. This will create 2 single strands of DNA. One strand is going to serve as a template to be paired with the complementary base pairs to replace the original strand of DNA. The other strand of DNA will serve as a template to make the RNA in the form of mRNA. It will use the complementary ribonucleotides. It will use ribonucleotides instead of deoxyribonucleotides because it will be making RNA. This means it will use uracil instead of thymine. When this is complete, the RNA is sent out into the cytoplasm, and the piece of DNA that was used as a template will be degraded and recycled for making new pieces of DNA in the future. Translation occurs in the cytoplasm. This is going to be the reading of the mRNA to make the protein. The ribosome will attach to the mRNA because it is the organelle able to read the entire genetic code. It will search along the mRNA and begin reading at the first AUG sequence it encounters. AUG is the codon that codes for a specific amino acid, and in this case it's going to be methionine. As far as we know, AUG serves as the start for all organisms. Each amino acid is going to be attached to a tRNA. On one end of the tRNA there is going to be an anticodon, which will be base pairs complementary to the codon. The tRNA is used to transfer the amino acid to the ribosome, and the ribosome will add the amino acid to the growing polypeptide chain. After the ribosome picks up the amino acid, it will move on to the next codon to read the next step. This will continue until it reaches a stop codon that tells the ribosome it has reached the end of the gene for the coding protein. As soon as one ribosome finishes with the codon, another ribosome may begin using the same mRNA set of instructions, so multiple proteins can be made from the same set of instructions. This is a table of codons used in translation. Along the left side you will see the first letter, which will be in the codon. Across the top gives you the second letter. This is just to help you follow where you could match the U for the second letter, and then you look in the square where the third letter will be. What you will see next to it will be a 3-letter abbreviation for the amino acid. So for AUG, we would first find A here along the left side, U across the top. When you come down to that box, there are 4 choices. Over on the right is where you find what the third codon is. So finding A, U, G, where all of those intersect, is going to be MET, which is the 3-letter abbreviation for the amino acid methionine. There are 3 of them that say stop: UAA, UAG, and UGA. These are going to be the stop codons that will terminate translation. They can be found right here. So for example, if you had CCG as your codon, you would look C on the left, C on the top, G is the third base pair, and where they cross is PRO, which is the 3-letter abbreviation for proline. There are several animations here that we're going to look at that are going to help in understanding this process. These animations are also going to be available to you outside of this presentation so you can watch them on your own. This first one is presented by the University of Nebraska. So we start with our cells. Protein synthesis begins inside the nucleus of a cell. Inside the nucleus, chromosomes are stored. Chromosomes contain the information to produce every protein the organism will need, encoded in DNA. The information is encoded in the sequence of nucleotides of the DNA double helix. The process of protein synthesis occurs in 2 steps. During the first step, called transcription, an mRNA copy of the gene is made, which carries the encoded information to the cytoplasm. Every gene has 3 regions that occur in the following order. The first is the promoter, which turns the gene on and off. The second region is the coding region, which encodes the information for which protein is to produce. The third region is the termination sequence, which signals the end of the gene. RNA polymerase molecules are present in the nucleus. This enzyme is responsible for reading the DNA code and building the mRNA strand. The promoter region of the gene acts like a light switch. It signals when to turn the gene on and off. The RNA polymerase molecule recognizes that the promoter is signaling to produce the protein. The RNA polymerase binds to the DNA strand and the DNA double helix unwinds. Once the RNA polymerase reaches the coding region, it reads a single strand. As it does this, it builds the mRNA copy. As the RNA polymerase reads each nucleotide, it brings in the complementary nucleotide and bonds them together, forming the mRNA strand. The nucleotide breaks free from the DNA, and the DNA folds back into a double helix. When the RNA polymerase reaches the termination sequence, the sequence causes the mRNA to fold back on itself. This prevents transcription from continuing, and both the RNA polymerase and mRNA strand fall off the DNA strand. This animation on translation is also produced by the University of Nebraska. The process of protein synthesis takes place in 2 steps. During the first step, called transcription, an mRNA copy is made of the DNA. The mRNA carries the encoded information in the DNA sequence from the nucleus to the cytoplasm, where the second step takes place. During this step, called translation, the mRNA strand is read and amino acids are linked together into a protein. The sequence of the mRNA is read by molecules present in the cytoplasm called ribosomes. The ribosome links together the amino acids also present in the cytoplasm. Zooming in on the mRNA strand, the individual nucleotides in the sequence can be seen. The ribosome reads the sequence 3 nucleotides at a time. Each group of 3 nucleotides is a single codon. A ribosome present in the cytoplasm attaches to the mRNA strand and begins reading the sequence. The ribosome looks for the start codon composed of the nucleotides AUG. This is where it will begin building the amino acid chain. The amino acids are transferred to the ribosome by tRNA molecules. tRNAs have an anticodon at one end and an amino acid at the other. The anticodon of the tRNA dictates which amino acid it carries to the ribosome. Each of the 20 amino acids in plants pair up between 1 and 4 anticodons. The tRNA structure is actually the RNA coiled up into the proper shape. The anticodon is a sequence of 3 nucleotides that are complementary to a codon. When a codon is read by the ribosome, a tRNA with a complementary anticodon temporarily binds into place. The ribosome reads the next codon, and another tRNA with a complementary anticodon binds. The ribosome promotes a chemical reaction to occur that joins the 2 amino acids together with a peptide bond. As the ribosome continues down the mRNA strand, the first tRNA falls off, leaving its amino acid. The tRNA matching to the next codon binds to the ribosome and the amino acids bond. This process continues until the ribosome reads the stop codon. This codon indicates the end of a gene. The last tRNA falls off the ribosome and the ribosome falls off the mRNA strand. Although the chain shown here only has 5 amino acids, in reality an amino acid chain is usually composed of hundreds of amino acids. The amino acid chain folds up into a 3-dimensional structure dictated by the order of the amino acids. This unique structure gives each protein its unique function and allows it to do work. During translation, multiple ribosomes can read the mRNA strand at a single time, producing larger quantities of the protein.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the 2 parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown. DNA replication begins when helicase unwinds a segment of the DNA and breaks the hydrogen bonds between the two complementary strands of DNA. DNA polymerase can only add new nucleotides to a free 3' end of a growing chain. Synthesis of one strand of the DNA, called the leading strand, proceeds continuously in the 5' to 3' direction. Synthesis of the complementary strand, called the lagging strand, is more complex. DNA polymerase can add new deoxyribonucleotides only to a free 3' OH. To provide a free 3' OH starting point on the lagging strand, RNA primase attaches to the DNA and synthesizes a short RNA primer. DNA polymerase III then adds deoxyribonucleotides to the 3' end of the RNA primer. DNA polymerase I replaces DNA polymerase III, removes the RNA, and replaces it with DNA. Finally, the enzyme DNA ligase forms a phosphodiester bond between the 3' OH of the growing strand and the 5' phosphate in front of it. During DNA replication, the leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously.
This next animation will show protein synthesis. This next one is going to show translation. There will be additional links you may follow for more information watching these. I would encourage you to watch as many different animations and read this in several different sources. The more you go through this information, the more it will make sense. Please be patient with yourself, though. It does usually take multiple attempts of looking at it to understand protein synthesis. The following 2 pages are information that I would strongly recommend reading. The first one is going to be on transcription. The second one will be on translation. After the reading, there is an activity here, a NOVA sequence, that you can go in and practice sequencing DNA online. Transcription and translation occur very fast in the cell and are actually very accurate processes. Sometimes, at the speed at which they occur, mistakes will occur. Mutations are going to be changes in the genetic information. These can occur as a result of environmental causes or random error. These worksheets are here for you to print out and do. These are not to be turned in. These are just to help with your understanding of this process.